This is the remarkable Dr. Frank Scheer. Our paths first intertwined in an unexpected place, Antalya, Turkey, during a pioneering translational chronobiology conference. Back then, long before the accolades of the Nobel Prize and the profound exploration of applying circadian clocks to clinical medicine, we found ourselves at this very remote conference where people weren't really sure what the field was about. And yet this encounter sparked shared excitement for a field and all kinds of unimaginable possibilities at the time, which we've all been working on since. So today we find ourselves standing at the threshold of a new frontier called circadian medicine, with applications in lots of areas. For us, it's cardiovascular health and disease. Dr. Scheer's presence here is a testament to his unwavering dedication and expertise, and I extend my deepest gratitude to him for gracing us with his visit to Guelph and for generously agreeing to deliver both this short talk to both of us as well as to members, familiar faces in the CSC meeting tomorrow. So by way of biography, Dr. Scheer is a professor of medicine at Harvard Medical School and a senior neuroscientist at Brigham and Women's Hospital. He has an H-index of 75, and in just the past 5 years, his work has garnered over 13,000 citations, showcasing his far-reaching implications and his groundbreaking studies. Dr. Scheer's visionary leadership extends to the co-founding and directorship of the esteemed Medical Chronobiology Program at Brigham and Women's Hospital. This interdisciplinary research program serves as a hub for translational researchers in sleep and circadian biology, aiming to unravel the underlying mechanisms driving time-dependent variations in disease severity. So Frank, we have small tokens for you, but we will give them to you at your talk tomorrow. And thank you. So this is for you, and that's for your talk.
Great. Well, thank you so much, Tammy, for that kind introduction and for inviting me to speak here. I'm really so excited. This is such a wonderful conference, and I love the idea of bringing together the chronobiology folks together with the cardiovascular folks. That's really exciting. So, yeah, I'm really honored to speak here today, and I couldn't have had a better speaker right before me than Martin Young. So you kind of gave my talk already, but so thank you for that. So indeed, you know, my introduction of the circadian system can be quite short. And I do want to point out this one, I think, quite remarkable observation that Nathaniel Kleitman made in these mammoth caves that Martin just spoke about, which is that they went into these mammoth caves and they were in a time-free environment in a way, and so then they painstakingly with each other, they recorded each other's core body temperature as some marker of physiology. And so what they found was that if you look at the bottom panel, you can see what you would see in most of you, which is that core temperature is high when you're awake and it's low when you're asleep. Sleep here is indicated by the gray area. But what's remarkable, if you look at the middle panel, so the 28-hour sleep-wake cycle that they put themselves on, you see something quite different. So, and I think this is quite fundamental to the understanding of circadian biology, which is that if it were just purely driven by your behaviors, you would anticipate that temperature would rise when you're awake and would decline when you're asleep. But that's not the full story. Yes, there is an effect of behaviors, but it's not the full story. And I think this becomes even more clear if I just, cartoonishly draw 24-hour dotted lines here. You can see that actually there is this underlying rhythmicity still persisting at a rhythm of 24 hours, in addition to the influence of the behaviors. And such that at the beginning, when people are in phase, the amplitude is large. In the middle, the amplitude becomes really small because there are two opposing influences between the behavioral cycle and the circadian cycle. and then they become re-entrained again, or in alignment, I should say, again. And so this was really one of the first discoveries in the end of the 1938 of the existence of circadian biology in humans. But it wasn't understood at that time what is driving this, what is enabling humans to express this. And so this had also been shown in plants about 300 years ago, that plants can open and close their leaves in the absence of an external time cues. So, and I make one big apology because, as you will see, the heart is missing. So this is from a review where we're focusing on metabolism, but imagine the heart there and the lungs and the vascular system. But the point I want to make is that and as Martin so beautifully already described, in the early 70s, the discovery was made, where is this coming from? This ability to generate circadian rhythms in the absence of either behavioral or environmental factors. And so what they did is they placed lesions in different brain areas and found that one brain area, if you lesion that, the animal becomes arrhythmic, and that was the suprachiasmatic nucleus. Now, suprachiasmatic is because it's on top of the optic chiasm, where the optic nerves cross, from which, of course, light input is the most important time cue, or zeitgeber. And so it showed, these lesion studies, that the SCN is necessary for the animal to show at least behavioral rhythmicity. But also, if you place this structure, this nucleus, in a petri dish, it happily ticks along with a rhythm of about 24 hours. So it's not only necessary, it's also sufficient. So for the next about 20 years or so, from the early 70s, it was understood that we have one clock and one clock only. But then in the 90s it was discovered that we have basically the same molecular machinery residing in virtually every cell and tissue throughout our body, and not just the metabolic tissue. And so this, as Martin already described, is generated primarily by the transcription, translation, feedback loop of these core clock genes that enable cell autonomous oscillations. So even a single skin cell, a heart cell, a fat cell, you put it in a petri dish, it can tick along with a rhythm of about 24 hours. So suddenly the system became a lot more complex because we didn't have one clock, but we had this multi-oscillator system. Now this would be fine and well if it weren't that, as Martin already explained, these different oscillators are not listening to the same time cues necessarily. And let me also first just say two things. One is that the central clock, the SCN, uses neural, endocrine, and temperature signals to do two things. One, for example, think about the heart. Autonomic nervous system regulation is modulated by the circadian system, as we'll hear in a little bit. And so the SCN outputs through neural and hormonal control influence physiology directly. And second, it can also synchronize or help synchronize these peripheral oscillators throughout the body. So there's direct physiological influence, and then there is influence indirectly through modulation of the peripheral molecular machinery. So, and the system is more complex, as I said, because these different zeitgebers affect the different oscillators throughout the body differentially. So light is the most potent zeitgeber to the central clock, but in addition to receiving information from the SCN, these peripheral tissues also are impacted by our behaviors, such as especially our eating fasting cycle. And there's also evidence for the effects of, for example, exercise. And so imagine now you are here in the Eastern time zone, but if your eating fasting cycle is misaligned, your liver might actually be somewhere in Tokyo. And so this understanding of what we call internal circadian misalignment is where your different oscillators are not all residing in the same time zone. And so the understanding, as Martin explained, is that this conflict in time signaling deriving from the SCN and within these peripheral oscillators may underlie some of the adverse consequences, although at this time this is still very much a working model. Now, the understanding of the regulation of the circadian system of our physiology also has wide-reaching effects way beyond just regulation of sleep. Yes, sleep is regulated by both homeostatic and circadian effects, but most of our physiology is affected by our circadian system to some degree. And of course, there's great interest into the relevance for pathology as well. And I will keep this short. So in my talk, I will briefly talk about some evidence towards the regulation by the circadian system of cardiovascular function, the interaction with behaviors on cardiovascular function, and circadian misalignment, and if there's something we can do about that. So I don't need to dwell on this. We know that there are strong day-night rhythms and serious cardiovascular events. And what they all share is that they have a peak approximately between 6 and a.m. and noon, so in the morning hours. And most of the work that had been done so far, up until chronobiologists became interested in this, was looking at behavioral triggers that may help explain this. Because of course, we're waking up, we're transitioning from supine to an upright body posture, etc. So, but could it be that the internal circadian system together would be influencing cardiovascular function and risk markers? And so this was work together with my former mentor, longtime colleague and friend Stephen Scheer, in which we had people come into the lab, live there in a time-free environment for 2 weeks, in which after 2 baseline days we enter them into a forced asynchrony protocol. So this is akin to the studies by Kleitman, except that they're now more controlled. And we keep people in dim light conditions so that we can extract the influence of the endogenous circadian system, because, as indicated by the dashed line here as a cartoon, the core body temperature under these conditions ticks along at the inherent rate of the central pacemaker, which is about 24.1 in women on average and 24.2 in men on average. And so what it allows us to do is we could then schedule test batteries to occur at all different circadian phases, and to not only look at circadian control, but also the interaction of the circadian system on the reactivity of the cardiovascular system to a standardized stressor. But if we just look at in a resting phase, are any cardiovascular risk parameters influenced by the circadian system? The answer is yes, and Martin already showed this very nicely, that PI1, plasminogen activator inhibitor 1, which suppresses fibrinolysis and therefore is a pro-thrombotic risk factor, shows a very large amplitude, tightly controlled, beautiful circadian rhythm with a peak at about 6 or 7 a.m. if you translate circadian phase to the average sleep-wake cycle in these participants. And what it means is that this is at least one factor that's very strongly regulated by the circadian system, independent of the influences of sleep-wake and rest activity, etc., and could therefore potentially be involved in the increased vulnerability in the morning hours. Now, I do love this title, Martin. Pie for breakfast, whether you like it or not. I think it really captures the essence of what we're trying to do, is to show that whatever you're doing, your circadian system is modulating your pie there. And so this was, I shared with Martin, so thank you, Martin, for showing this already, so I can be really brief here, just to say that we try to, first of all, categorize which parameters are actually under endogenous circadian control. Second, whether the timing might be consistent with increasing risk in the morning hours or not. And so the main message here really is that if we look at it, 2 things jump out. One is that PI1 is high at that time. You can consider this as regulation of fibrinolysis. So the fibrinolysis is really the brake on blood clotting. So the brake goes away because pi1 is high, and at the same time the platelets themselves are activated. So you're pressing on the gas pedal, releasing the brake, and so we think that this might be important in circadian regulation of a cardiovascular risk parameter. What about behavioral interactions? And just to show the same cartoon, so this is really what is the effect of a standardized cardiovascular challenge at different circadian phases. And what we did is we did exercise testing, 60% maximum heart rate for 15 minutes, 70 revolutions per minute, so standardized. And what we found was that the increase in sympathetic nervous system markers, plasma epinephrine and norepinephrine, are strongest in the circadian morning. At the same time that we also see the biggest withdrawal of markers of the parasympathetic nervous system. So cardiac vagal modulation markers based on heart rate variability. So again, we have this system, this regulation of at the circadian morning, an increase in the gas pedal and the release of the brake, if you like. The surprising thing is that, as Martin already nicely pointed out, is that the morning surge that people describe in blood pressure is not due to the circadian control. In fact, blood pressure is at its lowest in the circadian morning. But interestingly, we recently found that actually the recovery after the exercise is slower in the morning. We also looked at other stresses there as well. So from this, it seems that it's actually the autonomic nervous system in response to behavioral stressors that are under strong circadian control and may be important in the morning, contributing to the morning increased risk, because the increase in sympathetic nervous system is largest and the decrease in parasympathetic nervous system is largest at that time. So could it be that, what kind of evidence that we have that blood clotting and autonomic nervous system are involved in the morning peak in serious adverse events? Well, there's some evidence in the sense that if you give aspirin or if you give beta blockers, both can lead to a blunting of this day-night rhythm in serious adverse cardiovascular events. So suggesting that yes, maybe blood clotting and autonomic nervous system control are important in this matter. So the last topic to discuss is circadian misalignment. So, and of course the most obvious example is shift workers when they're working at night. Even permanent night workers are not typically entrained to their night work. So their circadian night is still in the night, but they're awake, and their circadian day is still during the day, even when they're sleeping. So this causes a mismatch between the internal body clock and the behavioral cycle. And so many studies have shown that shift work leads to or is associated with increased risk for cardiovascular disease. We and others have shown with experimental circadian misalignment, this is actually causal, so we can test causality in this study in the sense that we can look at cardiovascular risk parameters being impacted by experimental circadian misalignment that may in the long run help explain increased cardiovascular risk. But can we do something about this, and specifically can we use meal timing to counteract some of these adverse effects? So two inspirations for this, and actually, Tommy, one was yours, and one would almost think that Martin and I had been talking before on which slides we would show, but truly, Tommy, your work, I think, is so elegant. So the tau mutant hamster, the heterozygous tau mutants have a period, circadian cycle length, of about 22 hours. So when you keep them in 24-hour light-dark cycle, they increase cardiomyopathy, as Tammy has shown. But the critical study was to actually then place them in a 22-hour LD cycle and show you can prevent this. So really elegant, showing that it's really the resonance, if you like, or the alignment between the circadian system and in this case the light-dark cycle that is important for health. The other inspiration was work from the laboratory of Carolina Escobar in Mexico, who did this elegant study where they had rats scheduled to night shifts by putting them in very slow rotating drums. So they weren't exercising, they were just not able to sleep at particular times. And these animals became obese and they developed adverse metabolic consequences. And there what she could do is by restricting food access to their active phase, so dark phase in these nocturnal rodents, they could prevent the adverse effects. So our question then is, can we do this in humans? Can we, by manipulating food timing, prevent adverse effects in simulated night work conditions? So I'll talk a little bit more about this tomorrow, but I just want to highlight just some cardiovascular endpoints that we haven't published yet today. So this is a little bit complicated, and I will not go through it in too much detail, other than to point out that in the top raster plot, so people were in the lab for 2 weeks, and so from day 7 through 11 they were in a forced asynchrony protocol. In the top one, when they were sleeping and eating on a 28-hour cycle. But in the bottom one, which was the intervention protocol, our question was, if we restrict food timing to be synchronized with the central clock, so run on a 24-hour clock cycle, can we prevent adverse effects by this manipulation? So I'll use orange as a control protocol, so the one that we know has adverse effects, and blue the effects of the intervention. So what I'll show is the constant routine protocol, which is another gold standard circadian protocol at the start, so day 3 and 4, and then after the forced asynchrony. So is there a change in cardiovascular risk parameters from before to after the circadian misalignment? So that's shown here. So on the left is the control protocol, and on the right is the intervention protocol with time restricted to the 24-hour cycle to synchronize with the central clock. And what you can see, systolic blood pressure, diastolic, and heart rate, is that there's a relative decrease in blood pressure, both systolic and diastolic, with the intervention protocol. So in this way, meal timing was effective. It's interesting to see that there was not a change in blood pressure in the control protocol, which may have had to do with the fact that people were on a lower salt intake than maybe they had at home, even though we had gave them a fixed diet for 3 days prior to their admission to the laboratory. The other thing we found was that in the control protocol we saw an increase in pi1 and a decrease in heart rate markers of cardiac vagal modulation, so parasympathetic nervous system markers, and so this could be prevented by the intervention without a change in cortisol levels, at least not a significant change. So promise for the use of meal timing in this regard. So I think I'm running over time, but I just wanted to highlight some of these points very briefly to say that along the lines of what Martin just described, is that there is particular interventions that could be used or developed further that may help in preventing cardiovascular disease risk based on chronobiological understanding. And so what we did is we ordered them according to how strong the evidence already is today versus how what is more theoretical and would need more time to be developed. And so just a few examples are, for example, in the top left, the classical chronotherapy, so does timing matter of your medication, changes of behavioral timing. When should you exercise? And so Jingyi Cheng in my lab is looking at this. Could you induce specific phase shifting of particular organs versus other organs so that you don't have concurrence of these, for example, increase in platelet activation and pi1 all at the same time? Could you maybe shift the platelets? Use of melatonin, which we've published on the top right, and then eating, as I just described, as well as other phase-shifting agents, as Martin already discussed. So in summary, what I've hoped in this brief talk to explain and to excite you about is the role of the circadian system in cardiovascular function, for example, in blood clotting and in autonomic nervous system activity that may play a role in increasing cardiovascular risk in morning hours, as well as the promise, I think at this stage it really is, of the use of meal timing as a countermeasure. And Sachin, you'll talk about this as well, I'm sure. And so with that, I would like to thank all the collaborators, my team, funding, and you for your attention. Thank you. And I'm happy to take any questions if there is one.
I'm sorry,
Yes.
Hi, Dr. Nolan from University of Manitoba. I'm actually a nurse, a little bit of an outlier. I'm a newbie in your group, so I really enjoyed your talk. I was interested about your ideas around exercise and wondered whether it was also associated with temperature raising and then a more rapid fall. And
Right.
so I wondered if you had thoughts related to that or whether there were other mechanisms associated with the exercise that you thought were at play.
Yeah, that's an excellent question. So, and it reminds me your question about exercise effects on thermoregulation, and what comes to my mind is sleep, for example. So, Kurt Crouchy and colleagues in with Justice had these beautiful studies where they manipulated skin temperature, for example, or loaded the core, increased core temperature, which then subsequently led to increased heat dissipation through especially distal skin. And so what they found was that if you heat the distal skin, you can actually facilitate the sleep onset latency, so you can shorten people's sleep onset latency. And then this was followed up by work by Eus van Zomeren in Amsterdam, where they used this beautiful whole body suit with gloves and shoes and had tubing running through, so they could regulate distal, proximal skin and core temperature through warm or cold drinks. And also what they showed was that manipulating temperature, especially distal and proximal temperature, they could manipulate sleep. Now, and so this is actually being tested also in patients with insomnia and patients with cold extremities who have trouble sleeping. But that's maybe not what you're asking about. So
Not quite.
Yeah.
I mean, it's related, and it's been, that's sort of something that I would regularly recommend is a warm bath or a shower, but it's the drop in temperature that I think may help to, with the onset of sleep. But I was just wondering whether there was another factor that you were aware of in terms of exercise, whether it's, I don't know, changes beyond temperature that were impacting that change in relationship of the alignment.
of the alignment.
Well, you said, you could use maybe exercise at a particular time. The timing of exercise was going to be important in terms
I see. Yeah. So thanks for clarifying. Yeah, so the point I was trying to make is that if exercise at a particular time of the circadian cycle leads to a bigger change in cardiovascular risk marker, increase in cardiovascular risk marker, then maybe it's opportune not to exercise at that time. And so Jeannie Cheng has recently published in Diabetes Care work showing somewhat surprisingly maybe, but looking at the Framingham Heart Score, that this was correlational work in the Look Ahead trial, which is she looked at a 1-year follow-up and found that the patients who were exercising more in the morning hours, actually have lower framing heart, higher risk. So on the framing risk score. So that was the point I was trying to make, is that maybe there are certain times of day that exercise is better than not. But of course, exercise is always good. So if you have to choose do exercise, it just might be a manipulation of when it's better. Yeah. Yes.
We might have to hold questions just in the interest of time, but I'm sure
Happy to
Dr.
talk
Scheer is
afterwards.
happy to talk
Yes,
to you
yes, thank you.
afterwards.